Hello, I'm Bretton, one of our MCAT tutors here at Inspira Advantage, where we help students get accepted into medical school and other professional programs. And welcome back to MCAT Bites. Today, we're diving into the fascinating world of single-celled organisms and how molecular machinery help molecules assemble within them to create life at its most basic level. Let's start with some fundamental building blocks. Biomolecules. These include proteins, nucleic acids, lipids, and carbohydrates. In single-celled organisms, these molecules come together to form functional units that carry out all necessary life processes. Proteins, my favorite, are particularly crucial. They're formed when amino acids link together in polypeptide chains, which then fold into complex three-dimensional structures. This folding is essential for protein function in single-celled organisms. Proteins serve as enzymes, structural components, and play roles in cell signaling. Now, focusing on prokaryotic cells, like bacteria, these single-celled organisms lack membrane-bound organelles, but still have impressive molecular assemblies. The bacterial cell is enclosed by a cell membrane and often a cell wall. Inside, we find the cytoplasm, which contains ribosomes, plasmids, and a nucleoid. The nucleoid is a region where bacterial DNA is concentrated. Unlike eukaryotic cells, this genetic material isn't enclosed in a nuclear membrane. Instead, it's a single circular, does that look like a circle? Not really, but we call it a circular chromosome because it's all connected. That's highly condensed and associated with proteins. Ribosomes are another crucial assembly in prokaryotes. They are complexes of proteins and ribosomal RNA that synthesize proteins. In bacteria, the ribosomes are slightly smaller than those in eukaryotes, with a sedimentation coefficient of 70s instead of 80s. This brings up the question of what are the two subunits that make up the bacterial ribosome? Well, the total sedimentary size is 70, and the big ribosomal subunit is 50s, and the small one is 30s. Many bacteria also contain plasmids, which are small, again, circular DNA molecules separate from the main chromosome. These often carry genes that provide advantages like antibiotic resistance. And this is something we use in the lab to modify single-celled organisms and ask fun molecular biology questions. Now let's talk about cell envelope in prokaryotes. This typically consists of the cell membrane and cell wall, and in a gram-negative bacteria, an outer membrane as well. The cell wall is composed of peptidoglycan, a mesh-like polymer of sugars and amino acids. It's very, very sticky. Now, if you know this answer, awesome. If you don't, no worries, we're going to tell you in a second. But what is the main difference between gram-positive and gram-negative bacteria? Just list everything you know about this because the MCAT will ask. The answer is that gram-positive bacteria have a thick peptidoglycan layer, while gram-negative bacteria have a thin peptidoglycan layer, sandwiched between an inner and outer membrane. Some prokaryotes also have additional external structures, such as flagella, which are long whip-like structures used for movement. They're composed of the protein flagellin and are driven by a remarkable molecular motor at the base. Pili are shorter, hair-like appendages that can be used for attachment to surfaces of other cells. Some bacteria use specialized pili called sex pili for transfer of genetic material in a process called conjugation. We've already made a video on this. I recommend you check it out. Despite their simplicity, single-celled organisms have developed fascinating ways to sense and respond to their environment. Many use two-component signal transduction systems. These typically involve a sensor protein in the membrane that detects an environmental signal and a response regulator that changes gene expression accordingly. Understanding how these components and how they work together is crucial for grasping concepts of microbiology and cell biology to ensure that you get as many points possible on the biochem section of the MCAT. Thank you so much for watching our video, and I'll see you next time.